Hello, everyone, and welcome back. My name is Gavin Farrow. I'm your moderator for the Omics and Interventions in Geroscience research session. We are going to have 15-minute talks with 5 minutes for question and answer. Our first talk today is Michael Kuflisky, and he's going to tell us about pterostilbene, friend, foe, or frenemy. Thanks.
Hello, everyone. Thank you for being here today, and thank you for having me. As mentioned, the title of my talk is pterostilbene, friend, foe, or frenemy. Today, I'll be talking about some of my work on a compound, pterostilbene, that is currently on the market as an unregulated supplement. Before that, though, a little exposition is in order. So how did we get here? And no, I'm not talking about the 45th. I'm asking how we go from this to this. I came to the University of Washington to study the biology of aging. As an undergrad, I was absolutely captivated by the idea that the process of getting older, illustrated here by our illustrious 44th president, might not be an inevitable end state, but a knowable and potentially controllable process. I have a few diseases pictured here, and they all have one thing in common: our age. As we grow older, our chances of developing and risk of dying from these and other age-associated diseases increases. As your age is the common denominator among these disease states, current medical practice treats each of these states as individual diseases to be diagnosed and treated. But what if we could target that common denominator? Is it possible to intervene in the aging process to simultaneously ameliorate the onset and progression of these diseases? When I tell someone that I study the molecular biology of aging, I'm usually met with one of two responses. Wow, so you're trying to make people live longer? I wouldn't want to live to 160. Can you imagine how destroyed your body would be? Or more commonly, so you like to make those anti-aging creams? Aside from the popular notion of preserving a youthful appearance, there's a lot we might be able to gain from successful aging interventions. There is also potential to increase what's called health span, with implications for individuals and societies around the globe. You can think of health span as the amount of time during your lifespan that you remain in good health. Here in blue, you live in good health until late life, when you begin to experience a decline in health and develop several age-related diseases. During these years, you may be unable to work regularly, may need to be cared for, and may develop a number of age-associated diseases that further jeopardize your health. In a scenario with an intervention that is able to increase health span, you would enjoy more years in good health, allowing for increased quality of life for the individual and potentially reduce strain on societies that must cope with healthcare costs associated with aging populations. To get to that holy grail of geroscience, though, we must first develop a better understanding of how the aging process progresses to facilitate the identification of molecular targets capable of modulating lifespan and health span. One promising target is the mechanistic target of rapamycin. This serine threonine kinase acts as a master regulator of cell growth and proliferation, linking disparate signals within and without the cell to processes controlling cell growth and fate. This regulated mTOR signaling is also implicated in several different human cancers, such as lymphoangioleiomyomatosis and breast cancer. mTOR gets really interesting in the context of aging. Its inhibition via chemical, genetic, or dietary alteration has been shown to increase lifespan in every model organism tested to date. It's emerged as an important target in modulating lifespan, with many groups working to identify chemical inhibitors that may serve as lifespan or health span extending therapies. I work with the yeast Saccharomyces cerevisiae. In this system, there are two TOR genes, TOR1 and TOR2, which arose during a genome duplication event. The gene products are used in two functionally distinct complexes, TOR complex 1, pictured here, and TOR complex 2. TOR is an essential gene, and most other eukaryotes only have a single copy. Because yeast have two, deletion studies are possible to allow for the elucidation of its effects. By deleting TOR1 in yeast, we can interrogate TOR complex 1 activity without completely abrogating it. Rapamycin is an extraordinarily specific and potent inhibitor of the mTOR, with nanomolar concentrations being sufficient to inhibit yeast growth. It does this by first binding to the proline isomerase FPR1 in yeast, or FKBP12 in humans. The rapamycin FPR1 complex then binds to a specific motif on the TOR protein, both inhibiting substrate recruitment and sterically occluding the active site. To me, the specificity of this complex is remarkable. Tacrolimus, another macrolide lactone, also binds FPR1, forming an inhibitory complex that inhibits the serine threonine phosphatase calcineurin, bringing to mind that changes in the structure of the drug can bring about totally different targets. Indeed, we've tested structurally altered TOR inhibitors from collaborators that end up having no TOR inhibitory effect. Before moving on, I want to familiarize you with yeast growth kinetics, which you'll be seeing a lot of. These curves are generated by measuring the optical density of liquid cultures over time. Using an automated plate reader, I can produce high-definition growth curves that can provide a wealth of information. Nominal growth curves for yeast cells of the typical lab strains in a fermentable carbon source are characterized by 3 distinct growth phases. During the lag phase, cells acclimate to the environment, sensing what nutrients are available and prepare for intake and utilization of the detected nutrients. In the presence of a fermentable carbon source, like glucose, the cells begin to ferment and biomass is quickly accumulated, resulting in a sharp increase in optical density. Once the cells exhaust the fermentable carbon source, the dioxic shift occurs, wherein they prepare to shift their metabolic strategy from fermentation to respiration. During the respiratory phase, biomass continues to accumulate at a slower rate. There are many ways these growth kinetics can help us to understand how a compound might be affecting the cells. We leverage this to develop an assay that allows us to query the TOR inhibitory status of a compound using comparative growth kinetics. We can also, sorry, there's some stomping on my roof. We can also observe effects on respiration. All of this information from measuring the optical density of culture. So this assay utilizes a wild type strain and 2 mutant strains. The TOR1 deletion mutant strain has 1 of 2 TOR genes disrupted. This results in a hypersensitivity to TOR perturbations. This strain also displays no growth defect in normal conditions. The FPR1 deletion strain here on the right lacks the proline isomerase I mentioned earlier that is required for rapamycin to inhibit TOR. This strain is completely insensitive to rapamycin and its analogs. TOR status as a central regulator of cell growth and proliferation makes this assay possible. Chemical inhibition of TOR signaling results in observable culture growth inhibition. Using comparative growth kinetics of the strains I mentioned, we can infer the TOR inhibitory status of a compound. Here's a look at signatures of different types of TOR inhibitors in this assay. Here's the signature of rapamycin and other FPR1 dependent inhibitors. They must first bind to FPR1, then bind to TOR. As concentration increases, we see a dose-dependent suppression of growth in the wild type in white and the hypersensitive TOR1 deletion mutant in red. The deletion of the FPR1 gene renders this strain insensitive to TOR inhibitors that require FPR1 for their function. So note here that the FPR1 deletion mutant in gray remains unaffected at any dose. FPR1 independent inhibitors inhibit TOR signaling through a mechanism distinct from FPR1 dependent inhibitors. Here, the ATP competitive inhibitor TORN1 produces a similar response, wherein the hypersensitive TOR1 deletion strain is hit harder than the rest at low doses. As dose increases, the FPR1 deletion strain also succumbs to growth suppression. It was actually a surprise to us that this assay would be able to not only detect TOR inhibitors, but discriminate between different classes of TOR inhibitors, giving us information on likely mechanisms of action. As I mentioned earlier, rapamycin is a potent inhibitor of TOR signaling capable of inducing a growth suppressive effect at nanomolar concentrations. Rapamycin and its analogs or rapalogs are the only compounds I have ever seen result in this flattening of the yeast growth curve. This effect on growth persists, a slow but steady and apparently linear increase in biomass regardless of which stage of metabolism the culture is supposed to be in. Pterostilbene also displays some interesting growth kinetics that I'll get into in a bit. Speaking of pterostilbene, it's a natural analog of resveratrol found in various foods like blueberries and almonds. It displays enhanced bioavailability relative to other stilbenes, and this is potentially due to these methoxy groups making it lipophilic enough to ease transport into cells, resulting in a higher absorption rate. Pterostilbene is available as an unregulated supplement and marketed as a general health anti-aging supplement. Some people also use it as a nootropic or compound that boosts brain performance. In our TOR assay, there is a clear dose-dependent response, but we don't see the signature of any mTOR inhibitor. The TOR1 deletion strain in red is being suppressed to a greater degree than the other strains at any dose. Pterostilbene at high doses appears to be cytotoxic, completely flatlining growth. We've also tested pterostilbene's effect on yeast replicative lifespan and see a dose-dependent response. Lower doses provide the largest extension with reduced benefits as dose increases. Between 60 and 80 micromolar, the benefit is completely lost with a marked decrease in lifespan at 80 micromolar. So pterostilbene suppresses growth in yeast, or pterostilbene appears to suppress growth in yeast independently of mTOR and is also able to increase yeast replicative lifespan. My colleague Mitchell Lee, who you'll hear talk later, a graduate student in the lab at the time, now a postdoc, decided to try a combination treatment of rapamycin and pterostilbene to see how outgrowth might be affected. We were surprised to see that pterostilbene completely abrogates rapamycin-mediated growth suppression. It's as if the cells don't even see the rapamycin. Here in the wild type cells, we see control cells growing normally. At 80 micromolar, pterostilbene suppresses growth as expected. 5.5 nanomolar rapamycin produces that slow but steady growth curve. In the combined treatment, we see growth kinetics that don't suggest a synergistic growth suppression, but what appears to be a complete abrogation of the rapamycin-mediated growth suppression. This is also true in the hypersensitive TOR1 deletion mutant. As I mentioned, rapamycin is a potent TOR inhibitor. If it's working, we expect to see growth suppression. We wondered if this effect might be due to a drug efflux response. Here, I tested the protein synthesis inhibitor cyclohexamide in combination with rapamycin. The wild-type control grows normally with the distinct growth phases visible in a 24-hour window. Rapamycin-treated wild-type growth is slightly suppressed. The protein synthesis inhibitor cyclohexamide suppresses growth even further on its own, and the combination works to synergistically suppress growth, and this is also true at higher doses of rapamycin and cyclohexamide. And true too for the TOR1 deletion mutant. So this synergistic effect on growth is in stark contrast to what we see with pterostilbene. We know that pterostilbene suppresses growth independently of TOR. We know that it can increase yeast replicative lifespan and that it seems to be able to erase rapamycin's effect on growth. In initial experiments with pterostilbene at high doses, it seemed cytotoxic as no biomass accumulation was ever observed. After serendipitously forgetting some culture tubes in an incubator for an extended period, I noticed some of the tubes began to get turbid and I realized I needed to increase the experiment time. On increasing experiment time, I found that the cells actually do begin to outgrow. And this high dose pterostilbene seems to be associated with this extended lag phase, which interested me because I had not observed such a pronounced lag phase extension with other compounds, and this flatlining after the dioxic shift. So the complete lack of biomass accumulation after the dioxic shift in treated cells was a surprise to me as well, and I wasn't sure what this phenotype was telling me at the time. Because the dioxic shift represents a metabolic shift in yeast from fermentation to respiration, I hypothesized that this phenotype may be a high-level indicator of respiratory capacity. To test the idea that the flatlining is associated with impaired respiratory capacity, I grew a row 0 strain of yeast in the bioscreen to observe its growth kinetics. Row 0 cells are cells with depleted mitochondrial DNA. This specific strain was generated by exposing cells to the known DNA intercalator ethidium bromide, which is an established method of generating respiratory deficient cells. In the bioscreen, I observed the post-dioxic shift flatlining in the row 0 cells, bolstering the idea that this phenotype is an indicator of impaired respiratory capacity. With this information, I began to wonder whether pterostilbene was inducing mitochondrial damage, and I also wanted to know if respiration deficiency is sufficient to confer resistance to pterostilbene. I conducted a dose response in wild-type yeast and row 0 cells. Each line here is actually 4 to 5 individual growth curves that happen to sit right on top of each other. They'll begin to separate as dose increases. White lines are controls for both wild-type and row 0. Just note the flatlining in the white lines to quickly identify row 0 cells. Blue is the tera-treated wild-type and red is the tera-treated row 0 cells. At 60 micromolar, pterostilbene slightly suppresses growth in both wild-type and row 0 cells. At 70 micromolar, this trend continues with both strains seemingly equally suppressed. At 80 micromolar, the wild-type treated cells in blue are growing slower than the row 0 control. At 90 micromolar, the wild type treated cells in blue are growing slower than both the row 0 control and the row 0 treated cells. And at 100 micromolar and beyond, this trend continues with wild type cells becoming increasingly suppressed relative to row 0 cells. Taken together, these data suggest that pterostilbene may induce mitochondrial damage or uncouple the mitochondrial membrane at higher doses. Mitochondrial deficiency alone doesn't appear to be sufficient to completely confer resistance to pterostilbene. However, reduced mitochondrial activity appears to confer some amount of resistance. So pterostilbene can abrogate rapamycin-mediated growth suppression, where other drug combos with rapamycin synergistically suppress growth. Pterostilbene can increase yeast replicative lifespan, and high-dose pterostilbene is associated with loss of respiratory capacity. So this growth profile phenocopies the growth kinetics of row 0 cells. And row 0 cells are resistant to pterostilbene-mediated growth suppression. Pterostilbene has multiple reported actions in vivo and in vitro, suggesting it's a promiscuous molecule. Among these is the finding that it inhibits ROS and inflammation, which may have an indirect effect on TOR signaling. As a compound found in foods like blueberries and almonds, pterostilbene seems to be a friend. However, the effect on yeast mitochondrial respiration at higher doses is potentially concerning. And with that, I'd like to thank my lab, my advisory committee, the Nathan Schock Centers, and my PhD program, Molecular Medicine and Mechanisms of Disease.
Hey, great talk, Michael. I had a question for you. Does rapamycin combination treatment also suppress lifespan extension that you see with pterostilbene?
So that's difficult to say. We found that rapamycin doesn't seem to last long enough on plates to allow for replicative lifespan analysis. So we've not done combination treatments on RLS for rapamycin.
Another online question for you. Do the OCH3 groups in pterostilbene disrupt the pharmacophore of rapamycin that might account for non-additivity of rapa and teri dosing?
I don't believe so. So if I understand your question, you're asking whether the methoxy groups on pterostilbene are disrupting rapamycin's ability to bind to its target and inhibit TOR signaling. The motif on the FPR1 protein and the TOR protein are incredibly specific for rapamycin. And I don't believe that pterostilbene would be able to interrupt that.
Thank you, Michael.
Thank you all.
Our next speaker today will be Kelly Harris, who will tell us about a natural mutator locus affecting germline mutagenesis in wild and laboratory mice.
Exciting way. So this is a picture that sort of shows motivation for a lot of our work in my lab. Basically, it's a picture of the sampling locations of various populations that were sequenced as part of the 1,000 Genomes Project. And they're color coded according to a statistic, which is the proportion of single nucleotide polymorphism segregating in that particular population. That's a specific type of variant. So every single nucleotide polymorphism has an ancestral and derived allele, and ACGRT base pair, and it also has neighboring base pairs on either side. And the identity of these can be sort of informative about what caused the mutation. And what this shows is that the proportion of variants that are this type, TCC to TTC, varies a lot across the world, from around 2% of variants in Southern Europe to 1.5% in East Asia. And that might not seem like a lot of variation, but since there's sort of millions of variants across the genome in each population, it's very unlikely to occur by chance. And it also isn't a pattern that you would expect to get with the normal evolutionary processes population geneticists think about, with sort of natural selection for variants that do certain things, or patterns of genetic drift. And instead, what it suggested to us was that the actual process of germline mutagenesis was evolving over these pretty short timescales of a few thousand years that have separated these populations. And it sort of led me in a direction of trying to study like what's going on with these, and like, do they have a connection to other processes that involve mutation accumulation, such as cancer risk and aging. So basically, we still don't know in the case of humans what's driving this pattern. We're sort of getting there faster with other organisms, as I'll describe. But sort of two big possibilities are something in the environment that mutates DNA, or something genetic, say a difference between populations in the efficacy of some DNA repair genes, say. And we sort of I mentioned we still don't know what's going on with that signature in humans, but my lab is turned in part to looking at laboratory organisms, because one of these variables, the environmental one, is easier to control. And this is a PCA of mutation spectra, which are these histograms of relative abundances of different variant types in a bunch of strains of laboratory mice. And what you can see here is that mice sort of have differences in the relative proportions of mutation types, just the way human populations do, despite the fact that all these mice have been reared for some time in a very controlled environment. So pretty uniform diet, stress, exposures to environments. And even, and the fact that they show sort of similar patterns to what we say in humans makes me think it's more likely all these patterns have a significant genetic component. And then there's even not really much difference between these mouse strains and the ages at reproduction, which is another thing that in theory could drive the differences in natural populations. And I have a picture here of sort of a fly in some ointment, because like a fun thing about this is that mutation accumulation in these lab strains is sort of an annoyance to people who want to use them to study other phenotypes, since they sort of change the strain genotype over time. But it's also sort of just this inevitable process as they're reared in the lab, and it ends up being a phenotype that's useful for us to study. And so what we wanted to do was sort of use sort of the oldest trick people do with things like lab mice, QTL mapping, where maybe you cross two strains and look for regions of the genome where a certain ancestry is associated with, say, a mutation spectrum phenotype. And we actually didn't have to do this experiment because it had already effectively been done, but not analyzed in the way that we wanted to analyze it. So back in the early 70s, Ben Taylor generated this collection called the B by D, BXD recombinant inbred mice, where the parents were one parent was the standard black 6 C57 BL6J mouse strain. The other was DBA2J, which is about as divergent from Black6 as European and African human populations. And they generated a bunch of F2 recombinant offspring and then inbred these lines by brother-sister mating sort of ever since. And these are useful for mapping a lot of tricky phenotypes, I think including aging-related ones where like you really want to minimize or control for effects of the environment. And that's why sort of 6 different strains have been generated over the past 50 years. And there's at this point about 150 of these, with the oldest ones being almost 200 generations old. And so this is really unique because it's a lot of mutations accumulated on a very constant genetic background. And basically every generation is more flies in the ointment that we can see at the end. And another insight from Beth Dumont's work at Jackson Labs is that these are the mutation spectra look like in the PCA I showed you earlier. And I've just circled the 2 parental strains to show they're different from each other and sort of the proportion of mutations from C to A and C to T, among other things. So they do have this phenotypic difference that we might hope to map. And then with a small consortium, we sequenced the genomes of all of these strains and then called basically variants that were private to exactly one strain and probably arose as mutations during their breeding in captivity. And my very talented former postdoc, Tom Sassani, led this work. And so the first sort of sanity check we did was to check that the older strains had accumulated more mutations than the younger ones, sort of this molecular clock that you'd expect. And just this for comparison is the number of mutations you get when you just sequence one child and their parents and count the variants. So in comparison, these strains are a much higher resolution portrait of what's going on with germline mutagenesis. And well, the first thing we did was to look for genome-wide hits associated with the total mutation rate, and we didn't find those. But we found a really significant one on chromosome 4 associated with just the rate of C to A mutations. And this affects the total mutation rate too, but just not with too small of a magnitude to show up as genome-wide significant. And it's even more apparent if you look at sort of the fraction of types that are of mutations that are C to A. And this ends up being sort of the principal component of spectrum variation in these mice. And it's a difference of the mice with the B haplotype from Black6, about 15% of their variants are the C to A type. And the other mice, it's about 20%. And so, of course, we wanted to figure out what gene might be responsible. And the QTL interval mapped by this experiment contains about 100 genes, but only 22 of them have sort of a good candidate high impact protein coding variant. And of these, there's really just one associated with DNA replication or response to DNA damage. So this was sort of our favorite candidate gene from the very beginning, this gene called MUT-YH, which is involved in the repair of reactive oxygen species damage, 8-oxo-G nucleotides. And that's really nice because C to A mutations are what are caused by this specific type of DNA damage. And sort of going beyond that, there have been other studies of mice showing that knocking out this gene produces a more severe C to A germline mutation phenotype, and that it even has the same distribution of sequence contexts as what we see in the BXG germlines. And there's also a connection to human somatic mutagenesis, where basically there exists some known variants in MUT-YH that cause heritable colorectal cancer syndromes. And these tumors from these patients have a C to A signature that also looks very similar to what we see here. And so this was sort of our first concrete support of this hypothesis that maybe there's a shared genetic architecture between variants that cause these germline mutation spectrum differences and ones that affect somatic mutagenesis and cancer. And we even used sort of a software for inferring cancer mutational signatures for those maybe familiar with that, and found that sort of this signature represented in pink, SBS 18, that's sort of the reactive oxygen species MUT-YH associated signature, and it almost perfectly separates the B and D strains. And then this is actually a picture from a recent study by Moore et al. From Inigo Martin Carina's group at Sanger, where they looked at accumulation of mutations in normal tissues during aging. And I'm just highlighting in their orange how SBS 18 was sort of a significant contributor there as well, especially to colorectal tissue, among others. So one thing we're sort of really interested in now is whether variation in this gene might sort of play a role, even people who don't have this sort of clinically elevated colorectal cancer risk, in sort of the way that this and maybe other tissues age. So we still haven't narrowed down sort of the exact genetic change within MUT-YH that we think causes the phenotype, because there's sort of 5 different protein coding differences between the B and D haplotype in this region, but sort of working on strategies for narrowing this down. But the cool thing is that all 5 of these variants are segregating in wild mice. So we were able to look up publicly available sequences of sort of the 3 wild subspecies that contributed to laboratory mice, Castaneus domesticus and musculus, and they all have variation in these 5 loci. So that suggests that this isn't just a weird phenotype that arose in the lab. It actually captures something that affects mice in nature. But another thing that we can see from this natural variation, especially doing a multiple sequence alignment of other species of the MUT-YH gene, is that actually the B haplotype version, the one with the lower mutation rate, appears derived, and the one with the higher mutation rate appears ancestral. And this is interesting sort of tentative support for this idea population geneticists call the drift barrier hypothesis, which sort of predicts that mutation rates should be difficult for evolution to sort of optimize all the way, because basically accumulating maybe a few extra germline mutations or like, you know, some extra somatic mutations at an older age is like not damaging enough to fitness to be very efficiently selected against, especially in organisms like mammals that don't that have like relatively few offspring. And so this still needs a lot of follow-up, but it was very surprising to us that maybe you could have a MUT-YH variant that like was in this suboptimized state for a long time, and you could have one mutation that it seemingly improves its function. And just so if anyone here is a model organism researcher who has old recombinant in red lines lying around, we think this is a really exciting sort of new use case for them to sequence them and look for these mutator phenotypes. So love for, you should sequence them or collaborate with us to do it. And I'll just briefly touch upon a different project in my lab where we found a similar mutator allele in yeast. So this was work in collaboration with my triad Dunham, when my great postdoc Pengyao Zhang, who's going to be on the job market this year. But basically what she did was find some natural strains of yeast that are enriched for C to A mutations in their rare variants. And she was able to find that these same strains like did have a de novo C to A mutator phenotype in the lab. And she also found that basically they have variation in OGG1, which is a different gene in the same pathway as MUT-YH that repairs reactive oxygen species damage. And so then what she did was clone the sort of standard lab variant of OGG1, as well as this mosaic beer alternate variant into plasmids, and transformed them into OGG1 knockout yeast, and found basically the natural variant with the excess C to A mutations has sort of an intermediate mutation rate in this experiment. So it sort of nicely validated that these yeast also have a natural mutator allele in this same pathway. So it could be, together with sort of the evidence in humans, it could be quite a widespread thing. And we just are interested in figuring out why this pathway seems to contain a lot of mutator variation, and which others might as well. And so I'll just conclude sort of with where we're at in terms of this emerging evidence for this pleiotropy, this shared genetic architecture among germline mutagenesis and cancer risk, and sort of the load of somatic mutations that accumulate over aging. So basically the work I presented from my lab, we showed that there's this natural MUT-YH variation that's responsible for this mild natural mutator phenotype in mice affecting the germline. And then that's really interesting in light of the fact that variants of the same genes appear to cause human cancer and human somatic mutations. And so we, of course, are thinking now about how to test whether the human variants also affect the soma, or sorry, whether the human variants also affect the germline and the mouse variants also affect the soma. And there's sort of complementary work that came out from sort of the same set of labs in Sanger, actually, that other genes associated with cancer risk may also have germline mutator phenotypes, which is really exciting. And just sort of overall, like we're trying to figure out strategies for discovering more of these genes. So we have more than just sort of this one anecdote of overlap in these processes in this one pathway. And it's a little difficult just because these mutator phenotypes require sort of special circumstances, like these mouse lines, to sort of show themselves really strongly, but we're working on other methods that might open up more of them. So with that, I'd just like to acknowledge my lab and our collaborators, and answer any questions.
We have some online questions.
Oh, great.
So the first question says, mutation rate between the germline and soma are very different within an organism. Is there an epigenetic difference or RNA protein expression difference between a somatic cell and germ cell at the MUT-YH locus?
That's a great question. So are there sort of expression differences at MUT-YH between somatic cells and germ cells? I think the short answer is yes. Like Tom looked a little bit into sort of expression patterns of MUT-YH and also tried to find like QTL variation, or basically that might control its expression in different tissues, and whether that could be relevant in the mice. And I think there were probably differences in expression might be responsible for some of the stuff from the figure I showed of just the load of C to A mutations being like a lot higher in, say, the colon than some other somatic tissues even. And yeah, like I imagine that expression or epigenetics could have something to do with that, or just the amount of reactive oxygen species damage that affects each tissue.
Another online question. Fascinating talk. There's an old argument, JBS Haldane, I think, that a germline mutator phenotype would be advantageous whenever the force of selection is especially strong, e.g. during a famine or drought or high predation. So there would be a long-term benefit to keeping polymorphism for mutation rate in the population.
Yeah, I think that's definitely true. That just there's some experimental evidence, mostly in things like bacteria, that like if the environment is really stressful, like a higher mutation rate could be advantageous. I tend to sort of believe that less for the more complex the organism gets, just because yeah. I guess increasing the mutation rate always seems like a less efficient way of getting your adaptation than just sort of mutating whatever pathway you need to get your adaptation in the first place. But I think, yeah, at least in the case of model organism or of unicellular organisms with really strong selection, like those dynamics could complicate it further, and maybe in vertebrates as well. Would never rule it out. But yeah, it's always, I think it's interesting, like there's that hypothesis, and then the drift barrier one, which just says like the high mutation rates are bad, but it's hard to optimize them, and they both have like a little bit qualitatively similar predictions sometimes.
One more online question for you. So cancer risk is increased by the mutator allele. Do you think there could be other phenotypes that are being affected that might be advantageous, which are maintaining this allele?
Yeah, the question is like, could this allele be doing other things besides affecting cancer risk and the germline mutation rate that are advantageous? And like I feel like if I had to put money on it, like I could be likely to believe that, because I mean, in terms of the early embryo, I think like it has to, like cell division has to be fast for a development to work right, and that could limit the efficiency of some repair processes, and that might be why sort of germline mutagenesis is at a higher rate sort of in certain phases of embryonic development. And like you could imagine just, yeah, other considerations like that, where just selection on some other trait or maintenance of some other trait just, yeah, causes these others to be sub-optimized.
Thank you so much for the talk.
Oh, thank you. Thanks again for the invitation.
Our next planned speaker, Mitchell Lee, is not able to join us today, so our next talk will actually be from Daniel Promislo.
Thank you. I'm actually giving Mitchell's talk for him. Can you hear me? So I want to start by thanking the organizers on behalf of Mitchell for giving him the opportunity to show his work. I will do the best I can presenting Mitchell's really exciting work. Any weaknesses in the presentation are mine and not Mitchell's. This is just a last minute pinch hit. And it's also a bit of opportunity for me to pay it forward. I went to a meeting about 25 years ago in Boulder, Colorado, and lost my voice, and my colleague Ellen Moore kindly gave my talk for me. So I'm very happy to give Mitchell's talk for him. Mitchell has been working in my lab on the question of how genetics determines the response to rapamycin, and this talk falls really beautifully in line with all of the talks that we've had today, talking about response to rapamycin and genetic variation. So thank you to the previous speakers for giving various parts of the background. So some key points for today's talk. There are, as you know, multiple interventions that we know about that can extend healthy lifespan or health span. But there are lots of questions, and one of those is that we don't fully understand how natural genetic variation impacts health span intervention efficacy. I thought that Kelly's beautiful talk just previously pointed out that we know a lot about, for example, C57 black 6 mice, and most of what we know about aging in mice comes from that and just a few other strains. We don't know a lot about the kind of natural genetic variation that exists in human populations. So what Mitchell has been doing is using a fruit fly model that captures that natural genetic variation and using that to try and identify genetic modifiers, not just of health span, but of the effect of intervention on health span, and trying to understand their downstream metabolic consequences. As this audience well knows, their age is a major predictor of just about every major cause of mortality in human populations. And as we've discovered over the past couple of years, the past year and a half, COVID is very much influenced by age. There are other factors as well, of course, but age has a dramatic impact on the outcome of infection, unfortunately, for many older populations. And there are multiple kinds of interventions that extend lifespan and health span. There are dietary interventions. Diet restriction has been shown for many, many decades to work in rodents and also in every model organism that we use in aging, including in non-human primates. This work showing that reduced caloric intake has quite a dramatic impact on lifespan and age-related phenotypes in macaques. All kinds of environmental factors. These are data from NDUFS4 mice. So this is a complex 1 mutation in the electron transport chain. And these NDUFS4 knockout mice have a very short lifespan, a matter of days, but you can see that under, so this is under normoxia. Under hypoxia, it gets even worse, but under hyperoxia, I'm sorry, but under hypoxia, when they reduce oxygen tension, you can see that these mice actually live considerably longer. And then, of course, as we've already heard about from a couple of speakers today, there are pharmacological interventions that can increase lifespan, and the work that Mitchell's done that I'm going to share with you today is all about an mTOR inhibitor. And in particular, rapamycin. You already got a beautiful introduction from Chris Sell's talk. Rapamycin, so-called because it comes from Rapa Nui, isolated from soil bacteria. And I can skip this part because you heard a beautiful introduction to many of the details of this pathway, both upstream and downstream from Chris Sell earlier today. The one point that I do want to make is that mTOR really is acting, it's sensing extracellular nutrients and then acting through two sort of decision points. The first, in the presence of extracellular nutrients, it's activating growth and protein synthesis. And then the second, in the absence of extracellular nutrients, it releases this inhibition that exists. And so in the absence of extracellular nutrients, we see upregulation of autophagy and stress resistance. And it's this pathway on the left that you see that's associated with this prolongation of many different age-related, health span-related traits. And what's exciting for us, and that you've already heard about for our field, is that you can downregulate mTOR not just by absence of extracellular nutrients, but also pharmacologically, and here in our case with rapamycin. And so rapamycin downregulates mTOR, and you get this increase in autophagy and stress resistance, among other things. There's certainly, people are well aware of the sex-specific effect. Many researchers have shown that, as you can see in the top right here, the effect of rapamycin on survival in mice shows a significant increase in lifespan in females. There's a significant increase in males, but it's not as great. Similarly, in fruit flies, this work published over a decade ago showing an increase in both females and males, but considerably greater in females. It's an open question whether this is a true intrinsic difference of the sexes in the response to rapamycin to TOR inhibition, or if there might be a different dose response in males versus females. Maybe, at least in the flies, we just haven't gotten to a high enough dose in our experiments, and the same could be true in mice. There's a little bit on genetic variation, surprisingly little. So we know quite a bit about genetic variation for the diet restriction response. We're not going to show that to you today, but Jim Nelson's work in mice, the Caberline group's work in yeast, our own group has shown in flies, in collaboration with Pankaj Kapadia at the Buck, that there's considerable genetic variation for the diet restriction response. Surprisingly little known about genetic variation for the response to rapamycin and its effects on health span. We do see here in a SOD1 mutant that rapamycin actually reduces lifespan. Also in this DB mouse, rapamycin decreases lifespan. So there's a little bit of information that's known. And similarly, this work from Ben Harrison from a decade ago, Ben is now in our lab, showed that there was no beneficial effect of rapamycin in female flies, and at higher doses it was toxic. Similarly, Mitchell has some unpublished data showing a lack of response both in a wild type fly model as well as in a tauopathy model. So a little bit known on genetic variation, but not much, and it's really that gap that Mitchell has filled with his work that we're going to present to you today. So the question is, how does genetic variation, especially natural variation, the kind of natural variation that Kelly was just talking about previously in humans, we're interested in that natural variation in fly populations and how that impacts the response to rapamycin. To do this, we're using a population called the DGRP, or Drosophila Genetic Reference Panel. These were a collection of wild female flies that were caught in the Raleigh Farmers Market by Trudy McKay, and she then, in her lab, had them inbred for 20 generations with full SIV mating, brother-sister mating. And so after 20 generations, they were highly inbred, and over 200 of those inbred lines were then sequenced. There are about 4 million variants, about 2 million that segregate at least 5% frequency in the population for the minor allele. And all these lines are maintained and publicly available and are now in labs all around the world, including here at the University of Washington. So after all of this inbreeding, we can measure all of these lines for any trait of interest. And thank you, Dr. Harris, for your presentation, so I don't have to go into the details of how we use that to map complex traits. But I do want to tell you about the particular trait that we're interested in. So of course, we're interested in aging. That's what we work on in our lab, and those of you on this at this conference are interested in that. But we wanted to find a trait that would be highly repeatable, highly regulated. As we all know, age at death is an incredibly complex trait that's influenced by many, many different pathways. It's what we all work on, but it's a pretty messy trait. And so we decided to look for a simpler trait to measure genetic variation in the response to rapamycin. And fortunately, this work had come out back in 2000, showing that flies are very responsive to TOR inhibition. So genetic inhibition through knocking out TOR leads to much smaller size of larvae. So this is a fly larva, and you can see the mutants are smaller. And also a delay in the time from when the embryo is laid by the female fertilized egg to eclosion, when the fly comes out. And what this is showing is that both in the wild type and for the mutant, rapamycin treatment slows development time. And so that's what we thought would be really interesting to look at. So here's a fly life cycle, male and female mate. The female lays a fertilized egg, which about 1 day later hatches as a first instar larva. It then goes through 2 subsequent larval stages. The third instar larva then forms a puparium around itself, it metamorphoses, and then a few days later, magic happens and out comes an adult. So what we actually measured was the time from placing the eggs in the food to when this pupa appeared on the side of the vial. So time from egg to pupation. The food that the embryos were placed on either did or did not have rapamycin. And so every genotype was measured both in presence and absence of rapamycin. It was striking. It was really striking. We found highly sensitive strains. So here's one highly sensitive strain, average time to pupation 9 days in the control, 14 days in Rapa. And here's a sensitive strain. Average is 9 days across the board, 8.6 days. No difference at all. And here are all the lines that we looked at, 140 lines with means and standard error, 1 plus and minus 1 standard error. And you can see there are highly sensitive lines that show an increase of 4 to 6 or 7 days, and resistant lines that show no effect at all. Some might even show a little bit of the more rapid development, but mostly are from 0 to very great delay. So of course we wanted to map these. This is a Manhattan plot showing each one of these points is a single nucleotide polymorphism. We looked at about 2 million of these, showing the minus the log of the p-value across the 4 fly chromosomes. There are a few suggestive peaks, but nothing reaches a false discovery rate that is anything to write home about. So nothing is statistically significant, but we think there might still be some signals there. So we went from single SNPs to a set-based analysis. And in particular, we use a covariance association test. And so this is going gene by gene rather than SNP by SNP. So we define all the features at the level of a gene, and we create 2 models. One is a model that takes all the polymorphisms within that gene and asks how predictive of the feature, in our case, sensitivity to rapamycin, are all the features within that gene, and how does that compare to the prediction of all the other genes in the entire genome? And we get what's called a covariance association test score, CVAT score, where the R squared is how much of the variation in this trait can we explain? And then we determine significance using permutation tests. Happy to give you more details later. So initially we looked just at polymorphisms within the gene and nothing showed up at all. We then asked, well, what about plus or minus 500 base pairs upstream and downstream? And this one gene begins to emerge. And we then looked, well, how about plus or minus 1,000 base pairs? And this gene, CG6362, emerges with 1 million permutations, significant FDR. So what is this gene? We don't know, but if you look at the ENCODE, the mod ENCODE data, it is pretty interesting in terms of its expression patterns. It fits with this embryonic and pupal life stages. And this reminds me to tell you one of the interesting things that we don't yet know about it yet, but we're doing these experiments as we speak. We don't yet know whether this dramatic effect of rapamycin on development time and the dramatic variation that we see is due to a particular life stage, first, second, or third instar larvae, or it affects all life stages. So we're currently doing that work. So this particular gene points to embryonic developmental time as suggestive. Of course, we're looking upstream and downstream, so it could also be some of these other genes that are involved. Important to keep in mind, though, that there's very little linkage disequilibrium in flies, so it's unlikely that this is an effect on a gene that's highly upstream or downstream. So just to wrap up, natural populations show substantial differences in sensitivity. This is the greatest amount of variation for RAPA that I'm aware of, and it's one of the most, the cleanest mapping of traits in the DGRP that I know of. And That is due entirely to Mitchell's beautiful work. I wish he were here to be able to present it to you. There's this interesting result with regulatory regions flanking this particular gene that might be associated with sensitivity. Right now, we're doing metabolomics. Previous work from our lab has shown that often very weak genetic signals, due to the fact that there might be tens or hundreds of genes, each contributing a tiny fraction to the variation, can be amplified and discovered through metabolomics. So we are now comparing metabolomic profiles of sensitive and resistant lines. The work that Chris Sell showed us towards the end of his talk got me really excited. I was sending Slack messages to Mitchell about how we must be on the right track. We don't have the data yet, but we should have soon. And then the other question that we're excited to pursue is whether the effects that we see in development time are associated with lifespan and adult survival. Of course, that's the question that I'm sure many of you have here. And with that, Mitchell would like to acknowledge me for giving his very nicely put together presentation. It's his and not mine. Many collaborators and funders, and thank you for your attention, and I will stop sharing. And I can see that there are chats, but no questions yet. Is CG6362 expression altered in the resistant or sensitive lines? Great question. We have not yet done any RNA-seq work where we've started with the metabolomics, but it's certainly something that we'd like to work with. I haven't talked with Mitchell about it, but I'm guessing that he's already thinking about some qPCR for that particular gene.
Daniel, great talk. This is probably a naive question just about rapamycin, but is there a causal relationship between this developmental delay and the protective effect on lifespan, or are they just sort of two separate things caused by the rapamycin?
That's the $64,000 question, and that's the next set of experiments that we're working on, is to figure out whether they're causally related. There's certainly a lot of work that was done starting back in the 80s, Adam Chippendale, Michael Rose, looking at genetic covariation between development and lifespan. But in the case of the rapamycin pathway, I don't know the answer. Someone else might know, Chris or Matt or others on this call, on this conference might have some idea. I don't know.
Hi, Daniel. Tim Mackey from the Caberline Lab. You may not want to go down this rabbit hole, but in sensitive lines, if you let those pupae hatch, is there a non-50-50 sex ratio, given the differences, the observed differences on rapamycin between female and male flies?
Great question. We've actually also started doing some of that work. There's a graduate student in the lab, Renee Koig, who is really interested in that question. We don't yet know whether the effects are sex-specific in the larval stage, and that's something that we're interested to look at.
Thank you again for the talk.
Thank you.
Thank you. We're going to take a break until 10:30 for coffee and bio break, so see you all soon. Hello, everyone, and welcome back to the Omics and Interventions in Geroscience research session. Our next speaker will be Christine Santillis, seeking a conserved signature of aging using interspecies targeted mass spectrometry.
Spot on. Perfect. Okay. Okay, so quick question. Can everybody hear me okay? I'm so short. This is a... Yeah, I think that's probably going to be the best thing. Okay, any better? Perfect, okay. All right, so thank you very much for having me here today. So as Gavin already mentioned, I'm Christine Santillis. I'm a new postdoc in the McCoss lab here at the University of Washington. I just started about 2 months ago. So just to kind of give you a frame of reference for this talk, a lot of what I'm discussing is really kind of what I would like to do during my postdoc. It's a little light on personally generated data, but thank you very much for being here today. So the work that I would like to focus on is seeking a conserved signature of aging. So just a quick agenda. So I won't spend too much time on the background because as we've already seen, there's been a lot of great information on the concept of health span, but I'm going to talk a little bit about that and how it relates to the plasma proteome. My goal of seeking a conserved signature of aging using interspecies targeted mass spectrometry, how we will develop this targeted proteomics assay, how it will be translated to multiple species, and a quick closing. Okay, so my apologies if some of you have seen a talk of mine recently. This figure may look a little bit familiar, and that's because it is. Before I started my postdoc, I actually used to study metabolism in the eye, and we used to like to think about the eye as an ecosystem. But I think that metaphor can very strongly be applied to your body as a whole, because you have a lot of moving parts that need to work together in order to have a functioning human being. I think, there's a mouse, sorry about that. So I think your brain is, for obvious reasons, very important to direct the functions in your body. Your heart is necessary to keep you alive and move blood throughout your body for nutrients and oxygen. Liver is critical for metabolism. Skeletal muscle helps you move. And I think we need no explanation after the last 1.5 years of why our immune system is critically important. But in aging, a lot of these organs' functions start to decline. And so this kind of draws on the concept of health span, as has already been said, this part of your life that you are healthy. And you can kind of think about health span in your life as kind of a function of how well you function as an individual over time. Sorry, some of my slides got a little bit shifted here. Okay, so eventually, either with enough passage of time or just a loss of function due to a traumatic event or illness, you kind of cross over this threshold and enter this field of disability or dependence, where you can no longer perform the functions that you need or want to do without assistance. And I think a big goal in modern aging research is to really focus on health span, not necessarily improving how long we live, but what fraction of our life is healthy. And one of the things I'm really interested in trying to get at is, can we identify molecular predictors of health span in your blood? And I kind of already said this, but we really think that the circulating proteome in your blood is the best system to test this. One of the big reasons being blood can reflect the state of all of the tissues in your body. So just to take kind of a very small snapshot, a smattering, if you will, of the various molecules that we can consider in blood, proteins being my obvious favorite, working in a mass spectrometry lab. And just as an example, some of the things you can monitor in blood, hemoglobin A1C, you can track the progression of diabetes. C-reactive protein can serve as an indicator after a heart attack, what your risk of reoccurrence is. You have antibodies flowing through your blood that can relate to autoimmune diseases, previous infections. You have small molecules, such as uric acid and creatinine, that can relate to kidney function or the presence of a disease such as gout. And even your actual cells, so white blood cells, red blood cells, can relate to a variety of different diseases, such as current infection, cancer, or anemia. But I think what all of these molecules can be can be viewed as are individual measurement, are indicators of health overall. And so for the duration of this talk, I'm going to be talking about the plasma proteome a lot. And I just want to drill down a little bit into what this is. So I think most of us have probably had blood drawn at some point. And fun fact, the little colors on the caps of those tubes actually mean something. So if you take whole blood and isolate it in potassium EDTA, mix it up, incubate it, spin it down, you can separate individual components of your blood. So for starters, you get your red blood cells, white blood cells, and platelets, and then you're left with the liquid portion of your blood, which is called plasma, that includes various proteins, small molecules. It's the liquid portion of your blood. And so we really enjoy studying proteins because really proteins represent kind of a functional endpoint from your genome and as a response to the environment in which you live. And plasma in particular has a bunch of different important proteins that perform really important functions to a functional, healthy human being, including clotting, your immune system, metabolism, cholesterol transport. And it's really cool because, again, your blood circulates throughout all of your organs, so it can contain proteins from other organ systems in the body. And so this kind of brings me to this question. Can we identify a broader signature of aging in the blood? And other groups have started to look at this and have done some really elegant studies doing so. So just to show a couple of them here, highly recommend these papers, really interesting stuff. But one limitation to this work that I'm showing, all of it was done in humans. There are a couple papers where people have looked in mice, but none of them have looked in parallel in multiple species. And the question that I would ask is, we look at this and it's useful for humans, but it doesn't answer the question of whether this is if this is an evolutionary conserved function. Is this conserved in other species, any of these results? And so the current work that I just showed you, done in humans, was done with a variety of approaches. So affinity reagents, here I'm just using this term to refer to aptamer platforms or antibodies, RNA-seq, histology of the cells in the blood, looking at small molecules, and using untargeted proteomics. But there's some really important limitations to these approaches that have been used so far. I think the big one I can really hit on is these affinity reagents, because they're very specific to structural or sequence motifs, and their reactivity can be changed fairly easily. In some cases, even a single point mutation would be enough to render an antibody or aptamer useless. And also the other approaches described, there's trouble with reproducibility, quantification, and throughput. But the solution that we're proposing, or at least would love to test, is targeted proteomics, because it can get around these issues of throughput, reproducibility, issues with quantification, and it can get around the issue of changes in sequences, because we can highly tailor an assay specifically to each species that we want to look at. And so kind of our main hypothesis here, the lifespan of vertebrates can vary significantly, but we all share kind of the same loss of function as we age. And so if you take some really different species, humans for one, mice, which have been the workhorse, or at least a workhorse of aging research as far as vertebrates go for a long time, and now the domestic dog, which has a different lifespan and shares a similar environment to humans, can we identify a conserved signature of aging across all three of these species, which have different lifespans, that may serve as a conserved predictor of health span? Okay, so in order to do this, we need to collect plasma samples from young, middle-aged, and aged organisms. And so I think, I've already told you, we have humans, mice, and dogs that we're interested in looking at. And I want to just take a moment to call out our collaborators who've been very generous in providing plasma from mice and dogs. From mice, we're working with the Sweetwine, Marcinek, and Rabinovich labs. And for the dog aging project, they have already been kind enough to provide us with some preliminary dog plasma samples to get this work started. And one thing I do want to call out, we're not looking in individuals in either species, in any of these species of really advanced age, because we're not really looking for what is the exceptional organism that lives a really long time. We just want to get a general picture of what is the progression over the lifespan generally, rather than from the exceptionally long-lived. So first, we need to develop a targeted assay in humans. And then we can translate using these proteins that we've identified in people into these other species. And in order to do that, there's a couple things we need to keep in mind. So first, the fundamentals of this assay, we need to use undepleted plasma. I will get into more details on exactly what this means on the next slide, but just keep this in the back of your mind. The important thing again being this targeted proteomics assay can actually be translated to other species because we can target specific peptides unique to each species. We have the capability to quantify about 200 proteins in a single run. And these proteins can range from those indicative of organ function, immunoglobulins, clotting factors, apolipoproteins, the complement cascade, and we can also highly tailor this to take specific proteins from the literature known to be important in aging and lifespan and include those in our assay as well. And for each protein, we select 2 peptides to track it over time and monitor its abundance. And the most important things about these peptides, they need to respond well, they need to chromatograph well, and they need to be measured precisely. So essentially, we have a lot of ways of verifying that what we're looking at is real and it's consistent. Okay, so I mentioned to you on the last slide, we need to use undepleted plasma. And let me get into this a little bit. So one of the problems with doing proteomics compared to something like a sequencing approach is that in sequencing, you can amplify the signal of your DNA and kind of measure those really low abundance things. With proteins, we cannot do that. We have the proteins that we have in the sample, and we have to proceed with what we have. So plasma in particular has a bit of a limitation here. High abundance proteins make up the majority of signal in plasma. So this chart is just showing for the majority of the plasma proteome that we know of, kind of what is the scale of abundance. And when you look at this, it may not look like a huge difference, but for perspective, albumin is approximately 10 to the 10th in abundance, while the lowest concentration, IL-6, is a little under 10 to the 1st. So it's pretty significant. And a lot of other groups, including some of the studies I mentioned earlier, utilize something called depletion, where you use affinity reagents like antibody trap columns to remove a lot of these high abundance proteins up here in an effort to look at some of these lower abundance proteins a little bit easier. The problem with this is that in other species, currently that technology does not exist. We don't have antibody trap columns that work really well in mice, and we don't have any in dogs, at least to my knowledge. And even then, they don't work consistently, so we could be altering the proteome in ways we don't understand using a depletion method. So in order to make it comparable, we cannot use this approach. And for perspective, if you take something like albumin, which is approximately 10 to the 11th, you compare it to something like C-reactive protein, which, sorry, my box has got moved a little bit here. I have my mouse there. This is like trying to find a single 747 in the state of California. So, you know, you can find it on a map if you really want to spend the time looking for it. It's doable, but challenging. And if you try to go from albumin, again, shown here up at the top, to something like IL-6 at the very bottom, this is like finding a single cell of E. coli in the state of California. And so perhaps it's quite obvious that we are limited in what proteins we can examine using our approach. So something like IL-6 that's really low abundance, we will not be able to pick up with current technologies, hopefully in the future. All right, so knowing all of this, once we have these plasma samples, we can generate a reference pool where we make a pool from various individuals and try to get the most complex sample that would be most inclusive of all the plasma proteins that would be found in the proteomes of these different organisms, in this case, people, digest the protein into peptides, and then we use a highly fractionated set of runs, again, just trying to build up the most comprehensive view of the plasma proteome that we can using our experimental approach. We generate something called a peptide library, and we can use this to select for the best peptides that can quantify the proteins we are most interested in looking at. And once we have selected our proteins and peptides for quantitation, we go back to our original plasma samples, digest those from each of the individuals, and then we can quantify the amount of quantify the amount of proteins. In this particular case, the picture is apolipoprotein. But we can do this at the peptide level and at the protein level and get an idea of how much of each we have across age. All right, so I kind of showed you for people, that's how we would approach this. And again, back to my apolipoprotein A1 example, just showing you the sequence of this protein. You don't need to see the individual amino acids here. It's not super important, but I've highlighted a couple of peptides in purple here. And if I show you the sequence of the same protein in mice and dogs and highlight the same peptides, oh, sorry, I don't know why this turned purple. I think it'll still show you what you need to see here. If I show you the matched peptides between human, mice, and dogs, highlighted in yellow are residue changes between each of these species. So we cannot necessarily take the exact same peptide from humans and apply it to mice, but we can get very close and select peptides that respond well, chromatograph well, and are precise, if possible, in the same location even. But this is one example of where a mass spectrometry approach can be tailored to the species as opposed to relying on an affinity-based method. All right, so we've already talked about the approach we want to take, and I just wanted to mention the only things we're really changing to look in mice and dogs is the plasma samples that we start with and the final peptide library that we end up using. But otherwise, the approach is exactly the same. So in closing, we talked a little bit about why plasma is a great matrix to study the age proteome and what limitations are associated with it. We talked a little bit about previous work that other groups have done into looking for a signature of aging. And we talked about how mass spectrometry can incorporate throughput, reproducibility, and the cross-species potential that is currently lacking in the literature. And we're hoping that if we can identify a conserved signature of age across these species, maybe someday down the road, this could translate to a molecular predictor of health span. And I just want to take a minute to acknowledge my lab. Like I said, I've only been in the lab a couple of months, but it's a great group to work with. And especially Lillian, Eric, Deanna, and Julia have really helped me get started. And thank you very much to the Biological Mechanisms of Healthy Aging for the funding. And thank all of you for your time today.
Great talk. I'm actually going to start with a question. Have you considered incorporating plasma samples from known interventions that affect aging, like calorie restriction in mice, to see if it reverses or slows the signature you're looking for?
I think that would actually be a fabulous approach to take. I think it could even be used to quantify differences between mouse strains, because I mean, as was discussed earlier, there can often be genetic differences even between isogenic strains. So yes, I think that would be a great use of this assay, especially, and it's easy to tailor once you have that initial peptide library. Adding and removing proteins and changing the assay is very straightforward once you've generated that.
I have a question from Daniel Promislo. As a next step, are you interested in univariate approaches to identify single important molecules, or a multivariate approach to ask if the proteome overall can predict health span or other phenotypes?
That's a really good question. I mean, really, I'm interested in both. I must confess, I don't fully understand if we will have sufficient data points to use a multivariate approach. That's a personal lack of knowledge on my part. But both of those sound like a really great way a great direction to take this. Thank you.
Comparative biology is super interesting and it's super difficult, as you pointed out. Agents, etc.
Yeah.
And, I guess there's two things. One is quite a bit is known about the aging profile of humans. Making those, could you create a panel and interrogate specifically those changes in different species? And then the other thing is the terms of the panel of species that you have to try to expand those. I mean, that's kind of critical. How do you? There are people. Gubernova has done a little bit with different species of rodents and cells from those species and looking at perhaps some of the primate studies as a suggestion, and then
so if I understand correctly, would you mind repeating your first question?
A fundamental question was, specific panel
Yes.
to interrogate down.
Yes, and that's actually part of what we're hoping to do. So I left it very vague, but when I said proteins that are of interest in aging, that is one question I'd really like to ask, is that we have several really good studies that were done in human plasma, and they identified a variety of markers that were of interest and seem to change across lifespan in people. So we would want to include as many hits from those studies as we can, but we can also introduce other markers that may be related to senescence, things that are important to aging in that respect. So yes, that's definitely on the list of things to do. And as far as cross species, that's really, I think, are going to be our biggest challenge, is getting the sufficient sample that we need. And I think for mice and dogs, we're good to go, but expanding outside of that, I fully agree. Sample acquisition will be the trickiest part.
One more question from Alessandro. Considering that you'll be restricted to highly represented proteins, which are functional in plasma, would it be worth it to look for post-translational modifications, phosphorylation, acetylation, glycation even, that may change with age?
Okay, so the answer to that question, I have two things to that. So the first thing is, admittedly, our lab does not dabble too much in PTMs ourselves, but there's a really great community of mass spectrometry researchers at the University of Washington. Like the VN lab does a lot of great work in phosphoproteomics. And so yes, I think it would be really interesting to see if we can use the samples that we're getting to look at the phosphoproteome in tandem, because we're gonna have a good selection of ages. And I hope, if all goes well, also representation of sex. It would be really great to look at that.
Thank you again.
Thanks.
Our next talk is going to be by Ben Blue, telling us about advancing intervention screening in C. elegans through artificial intelligence and robotics.
Okay, start. Fingers crossed. Hey, can everyone hear me okay? Cool. All right, start timer. So thank you everyone, for the organizers and everyone for being here today, both in person and remotely. So again, my name is Ben Blue and I'm a PhD student in Matt Kaberlin's lab. So some of the other speakers here today have done a great job of kind of introducing the concept of geroscience and why we study the things that we do. But the thing that I kind of want to highlight that kind of forms a lot of the basis for our research is the necessity for high throughput longitudinal studies has established this really pivotal and important role for model systems, particularly invertebrate ones such as C. elegans, to provide a tool set for both mechanistic studies as well as drug discovery, and provides the scale that's necessary to do kind of the big foundational experiments that drives a lot of the research. And so my dream is kind of, this is a weird dream to have, but it's long been to have kind of a Fitbit for an invertebrate species, something that tells us not only the longevity, but also a variety of different health metrics, all in one experiment. And so this work that I'm starting to talk to you about today was, originally pivotably designed by an engineer in the lab, Jason Pitt. And so he developed this really, really exciting robotic tool called the WormBot. And so what this is you can see the video on the screen. It holds 12 well cell culture plates, and each of these has about 35 animals on them. So a single device has about 4,300 animals. And so I'm going to play that video again. And so you can see this high resolution camera is going through, and every 10 minutes, every single cohort of animals gets a high resolution image taken. And then on top of that, what's not shown here is that we go and we take a high resolution real-time video once a day. So looking at it 30 frames per second, where we can actually start to look at animal behavior. And so we've got a variety of these in lab. Some are sized for benchtop, some are sized for incubators, and we're generating terabytes of data every month. This is what the data actually looks like. And this is 3 control wells of animals given the vehicle, 3 wells that are treated. And so I'm gonna let this play for a second, but what you'll see, I think the first death that we have will happen up in the top left. One of the left-hand plates, and you'll see a red circle appear. And so this is when either myself or one of my students has gone through and determined that that animal right there has died, it has expired, and then we've annotated it manually. And so we'll let this play a little bit longer, and we'll start to see animals perish and die, and red circles start appearing. And so what's really cool about this is that we have not only the scale to do all of this in the laboratory, but also all of this is, in the interest of rigor and reproducibility, is highly recorded, and we can go back and reanalyze everything. And of course, this is just half of one of our cell culture plates. The real WormBot, fully functional, looks something like this, where we end up with 144 experiments running at a single time. And so from this, we've been running a whole host of experimental cohorts across a wide array of invertebrate lines. So fundamentally, a lot of my work has been looking at drug interventions and both wild type C. elegans as well as a grab bag of interesting disease models. So worms that are expressing tau or amyloid beta or your favorite protein of interest in geroscience, as well as doing genetic interventions. So taking, disease targets that we've identified through collaborations with computer scientists, and then knocking those down and seeing how it affects disease progression. One little case example of this is this was a natural product screen that we did, and this is just one WormBot. And so there's about 50 compounds represented here, looking at the percent change relative to the controls. And so what's really exciting about this is that one of the databases that's online for looking at previously published drug discovery in terms of aging is DrugAge, and they report a whole host of different compounds generated from publications. And on DrugAge, in the current build, as of about 2 weeks ago when I last looked, there's about 300 unique compounds tested in C. elegans. And so in one single, basically, cohort here, we've done about 1/6 of the reported data. And so this is really, really exciting for me to have the scale to do this. But I kind of want to take like a 2-minute detour and talk about something that I think is also really, really exciting and really, really important in science in general, but also the field of geroscience. And so all of the tools that we've built here and that we're using to collect all this data are all web enabled. And so, not only does this make things easier, particularly in the last year and a half, as sometimes lab access has been restricted, but to kind of broaden the scope of why do we do science and what do we do? So I've got a map up on there, and right now, Red Star in Seattle. And so a little bit about myself is I'm actually from this tiny, dinky little fishing town in Alaska called Homer. And so we're the halibut fishing capital of the world. And I want to preface that I think we were the ones that decided that. There was no committee that actually, that's us saying it. And so, as many of us were last year, I was working from home and thinking about it. And I saw a news article that they had moved the high school to all online learning. And so for me, when all of the biology that I was exposed to as a kid was all fishery science, and I really wanted to get away from fish, and it took me a long time to find biology as I was excited about. And so I wanted to try and do something cool. And so I went and I reached out to the high schoolers there, and I figured with this device that we've got here, and with Zoom and our classic hallmarks of aging kind of structure for organizing this, we went and we did about a 2.5-month biology aging class to high schoolers. And so part of this was them deciding what do they think in their lives that they've heard of is affecting their aging process. And so they picked a whole host of compounds. Some of them were things that you've probably tested in your own lab settings. A lot of different antioxidants. One student really wanted us to test Alaskan salmon. And I didn't think that would ship that well, so we didn't do that. And so then we set up these experiments, and then the students were able to go through and actually use our online tools. And this is taken from one of their lab reports. And they actually, I think this was glucose that they were interested in. And so I just kind of want to broadly talk about, the ability to create these tools. It shouldn't just be about our ability as researchers to work more efficiently, but also our ability to expand access to geroscience and the science that we do. Okay, so that's my 2-minute spiel on that. So going back to our big project here is that we're collecting just terabytes of data every month. And this analysis, even though it's partially automated or even allows us to be remotely doing it, it's very time intensive. And so coming back to this idea of a Fitbit, my goal is to take this and elevate this kind of functional screen to mine as much data as possible. So in the context of aging, we could look at, the percent of time spent on and off food as a measure of chemotaxis. The body morphology, growth rates indicate, metabolic function, and a whole host of other behavioral changes, and then contextualize all this within the metric that we all use day to day, which is total lifespan of an animal. And so we decided to try and take this and apply machine learning to this problem. And so we chose this neural network architecture called YOLO. And off the record, don't tell Matt, but I picked it just because of the name. I didn't pick it just because of the name. It stands for You Only Look Once. And so it was actually developed here at the University of Washington. And so what you can see is here is our early version of it going through identifying these animals in these very high resolution images. And so this is actually a really kind of cool computational problem because these images are higher resolution and the objects in them so small relative to it. It's actually like some interesting bit of methodology from just an AI standpoint that we actually were able to try and figure out and solve. And so with this pipeline, we wanted to also, as I've said, engage our community of scientists and volunteers. So we built this pipeline where images were collected from the WormBot. We used our early manually trained version of Worm YOLO to detect worms. And then I made a website here where students could log in from all over both the US, but also the world, and help us train this network in actually identifying and tracking these individuals longitudinally. And then, of course, all of that data is then brought back into the neural network and retrained. And so our pipeline that we're currently working with now has Worm YOLO identifying the animals. And then from that, we take the bounding box data and we track those animals longitudinally as soon as they begin to slow down in midlife. And then that tracking data is used to determine cessation of movement and changes in body morphology that indicate death. And so this is taking a cohort of about 400 animals across 19 different wells, comparing the human manual calls that you saw with us putting those red circles on with the neural network's output. And you can see that we've actually got this down to about 2 hours for the set of wild type worms that we partially use this data set to train the neural network on. But it's not just enough to do this in wild type. We're all interested in interventions. So in conjunction with the Mendenhall lab, we started looking at a couple of different genetic mutants. And so what you'll see here is each color represents a different genetic mutant of C. elegans, and then dashed is the manual calls, whereas green is the neural network. And well, you can see that even though in these strains that the neural network has never seen before, we are actually able to recapitulate the delta very accurately in the time of death for each of these strains. And then, of course, my interest is also in interventions. And so looking at this, we took a bunch of our historical data and processed it with the neural network. And, we're, within about a half a day of time of death difference. And so what's really cool about this is that historically, worm experiments were done by a researcher going into lab every day, poking an animal with a platinum wire and determining if it was alive or dead. And even if there's a little bit of variation here, that's still really, really exciting for us because it's smaller temporally than the actual change that a human experimenter would be able to do. But again, it's not just enough to look at longevity in the context of geroscience. We also really want to examine health span. So from those real-time movies, what we're able to do is then take them and look at how well the animals are moving at a given day. And so this is, the caveat to this is these images are taken once a day. But you can see each animal gets its own unique ID. And we're currently working on then correlating this data with the neural network. And so from that, we currently get out anywhere between 8 to about 10 to 12 different health metrics. So looking at the average speed of movement of the animal relative to the max speed, average and maximal rates of acceleration, the sinuosity of the track, all of these kind of help us contextualize when we're giving them a certain drug. Is it helping them maintain, the kind of anthropomorphized model of physiological health? Is it making them seem stressed out? Are they exhibiting fleeing behaviors? All of which is really, really helpful when doing drug screens that are in the thousands of drugs. And so then lastly, what we've just got online working now is looking at, we've built another layer to the neural network that takes the object of these animals and then segments out and actually lets us look at the specific underlying body morphology and subtle movements of the animal that is actually really, really exciting in the context of some of the Alzheimer's disease and neurodegeneration models that exhibit very, very subtle phenotypes that this should allow us to automatically measure. But it's still, this part of it is still a work in progress. What we just also have got going on is that we've also developed a fluorescent model that's going to let us actually monitor gene expression over time longitudinally in these populations. And so now we think about correlating all of these different metrics of health within the context of lifespan. We'll also then be able to look at several biomarkers of gene expression. And we're currently narrowing down the list of biomarkers that we hope to actually examine. So in summary, what we've kind of developed is what I hope to is a Fitbit for worms that lets us better marry the kind of anthropomorphized ideas of health and behavior within a high throughput invertebrate model. And so what's next for us? So currently we've got about 3 years. So hundreds of compounds, hundreds of genetic interventions that we're going back through. And because of this data, we're being all still stored in the lab, we're actually able to go through and remine it for all these novel phenotypes, in addition to the lifespan, which was originally measured. We've started in the last couple of months screening a FDA-approved library of over 1,900 compounds in both wild type and Alzheimer's disease animals. Given the Alzheimer's disease animals are actually very short, the phenotype is very short-lived in C. elegans in the specific model we're using, we actually hope to get through this library in the next 6 months, which would be really, really exciting. And then my goal is to take this and then use these metrics and start to predict early life biomarkers of longevity within this worm model. And lastly, applying this model to new models, applying this pipeline to new model systems. So we've started working with some other labs that do cell culture work or even some Drosophila work, trying to adapt this pipeline and all the work that we've done and see if we can't use it in other contexts. So with that, I have so many people to thank here, you know, my excellent committee and Matt and the rest of the Caberlin lab. And I really want to highlight that, you know, we've had a long history of working with students in the Caberlin lab. And I've highlighted students that, you know, all of these students that are on this list have been really excellent undergraduates. And those that have won Mary Gates Research Awards through their work with myself in the lab are highlighted as well. And so with that, I'd be really excited to take any questions that anyone might have. So thank you very much. I can get the ball rolling. I'm an Aquarius.
Hey, great talk, by the way. I was curious about the WormBot difference in estimated lifespan versus the manual.
Mm-hmm.
So in the data you showed, the WormBot mostly estimated that worms lived a longer lifespan. Is that true in most?
Across the board?
Yeah.
So that's a great question. So what we actually did to do that is we cross-referenced our manual annotations with the tracking data that we were able to get off of that and use that as kind of our training for the thresholding of when a worm is called dead. And so we had done that predominantly in N2, which is the main species of wild-type worms that almost everyone uses. Applying it to some of the different genetic backgrounds, we think that we'll have to adjust that thresholding a little bit in terms of getting, you know, the ground truth lifespan out. But in terms of getting a delta between control and treatment, or control strain and treated genetically modified strain, it seems like the delta is nearly 1-to-1 preserved. So I think it's just a metric of how long that thresholding amount is. Yeah.
Got a couple questions for you.
Yeah.
The first question is, do new eggs hatching in your assay cause problems?
So that's a great question. So for all the data that we've shown, we use a pretty standard treatment in C. elegans where we give them a FUDR, a compound that stops DNA synthesis. And so adult C. elegans are post-mitotic. And so given FUDR, they actually don't produce any eggs, and the eggs that, you know, might be kind of grandfathered in is the wrong word, but are still on the plates when the experiment starts, are hatch and, or, you know, die.
As a follow-up, does FUDR affect lifespan? Hasn't there been some papers on that?
Yes. So we know, this is a caveat of a lot of C. elegans work, is that we know FUDR is a hammer rather than a scalpel in terms of preventing progeny from being on a plate. And so there's great work by you know, Nikolai and the Mendenhall lab and other folks that have really looked at what are the actual like molecular effects of FUDR outside of what we're all using it for. And so there's other ways to get around this. You know, if we really there's this new technique of auxin-mediated sterility. You can use lines that are obligate outcrossers and then take off the males or females depending on what you want to use as well. But in the context of screening, we felt that FEDR is kind of well established in the community.
One more question. Hakimi has a protocol for very low dose FUDR 1 to 2 micromolar to suppress development rather than reproduction.
Well, I'll definitely look into that. Yeah. Okay.
Thank you again.
Thank you
Great
so
talk.
much.
Very
Appreciate
exciting. Our last speaker of this session is Kimberly Alonje. We're going to hear about age-related changes in hippocampal perineuronal net sulfation patterns.
I have to take my mask off or I fog my glasses up. So I get excited. Okay, so thank you for that introduction. My name is Kim Alonji, and I am an acting instructor at the University of Washington Medicine Diabetes Institute. And today I'm going to be sharing with you my research regarding perineural nets in the hippocampus during aging and development. So perineural nets are extracellular matrix structures that enmesh the soma and proximal axon and dendrites of critical neurons involved in memory and cognition. And we can visualize these nets through their distinctive honeycomb-like structures. As you can see, the honeycomb structures allows for the incoming synaptic batons to interact and form stable synapses with the neuron. And visualization is also possible through very traditional confocal microscopy, where you can see this net-like pattern even through histology imaging. What we're imaging is the chondroitin sulfate proteoglycans, which make up the lattice formation of the nets. We can image for the proteoglycan core protein in red, this would be aggrecan, or we can look at the attached glycan sugars known as the chondroitin sulfates, where we use the WFA lectin to recognize. My research really investigates the glycan portion of these nets, because the glycan portion is very heterogeneous in the mixture of the sulfated isomers incorporated into the polymer chains. Each isomer has a very distinct function biologically in the brain, and the relative abundance of each isomer throughout aging influences the brain's ability to maintain memory and cognitive functions. So, for example, in development is known as the developmental sulfation code. And this code is very high in very specific isomers, including the C isomer in yellow, which allows for brain plasticity and a softening of the brain during matrix and nerve circuit formation, and the D isomer that allows for neurite extension and regeneration. But when you move into the adult, you age into what is known as an adult sulfation code. And this code switches the isomer abundance from this C gold isomer to the A blue isomer, which is about matrix stiffening and decreased plasticity. It also will include dermatan sulfate to help stabilize those formed synapses. And then in both developmental stages, you can have isomers that play a functional role, including the non-sulfated isomer for diffusion of positively charged peptides, and also the E isomer, which has a role for negative neurite extension and development, but also stabilizes matrices in the adult. So the way I measure these different isomer sulfation codes is through tandem mass spectrometry. I use an enzyme called chondroitinase ABC, where we isolate the individual isomers off of our tissue of interest, and we can separate it through liquid chromatography, as seen on the x-axis. This is a time-dependent separation, and then the tandem mass spectrometry, which we assign a very specific channel for each isomer depending on its parental and daughter fragmentation ion patterns, which is shown on the left-hand side. And from this chromatogram, you can see that each of our 6 chondroitin and dermatan sulfate isomers can separate both on time and their assigned channel, which allows us to look at sulfation patterns in biological tissues. Here's an example of the extreme differences that happen in the sulfation patterns throughout development. This study was done in collaboration with Mike Gutman at the Department of Medicinal Chemistry here at University of Washington. We have a developmental series from postnatal POPs P7 all the way up into adult. When we look at our first isomer, the C isomer here in gold, which allows for plasticity of the brain during development, we see it's about 30% at P7 and then decreases into the juvenile stage and then finally reaches about 3% in the adult. Meanwhile, we have the matrix maturation isomer is lowest after birth at P7, which then increases throughout maturation to about 90% in the adult. And so this is a very yin-yang type of relationship when it comes to switching your developmental sulfation patterns to your adult sulfation pattern. But a lot of key questions remain when it comes to sulfation pattern changes in the brain. And that really comes down to, can we decipher these patterns on a more micro scale instead of the whole brain? For example, one question would be, are there interregional differences in the patterns? This would be an example of a gross anatomical difference between, let's say, the cortex and the hippocampus. A second question would be, are there intra-regional differences? And this would be differences between a singular region, such as the hippocampal CA1, 2, 3, and DG. So I'm going to answer the first question first, which is, are there any interregional differences in these sulfation patterns? And I chose the cortex and the hippocampus to investigate. In this example, I looked at the somatosensory barofield cortex here in the pink and compared it to the hippocampal region, not cortex, in the blue. And so I chose these areas because morphology-wise, they look very distinct in the way the PNNs were formed, where the somatosensory cortex is high in abundance and structured, while the hippocampal cortex is less structured and less abundant. I chose to look at a P21 mouse because I believe this mouse would show isomers that represent both development and adult and would be able to detect all of them. This initial investigation was done in collaboration with Nizam and Mirzadeh at the Barrow Neurological Institute. And so what we found is that there are vast and robust differences between different brain regions of the mouse in terms of their ability to mature over time. We find that when we look at our developmental A and C isomers, the hippocampus remains plastic for a much greater time than the somatosensory cortex. And this is observed by a greater ratio of that plastic C isomer gold bar compared to the somatosensory cortex, which had a lower abundance of C, but higher increase in the mature isomer. When we looked closer at the disulfide isomers, we saw a very similar pattern in terms of the isomer suggesting the hippocampus remains plastic for a longer amount of time during development. We see that the neurite extension isomer, which would promote neurocircuit formation, is higher in the hippocampus as seen in the green bar compared to the cortex. Conversely, we see that the cortex has higher disulfide isomers shown in the adult, including the dermatan sulfate for mature synapse formation, as well as the E unit, which would bind to semaphorins and stabilize mature PNN formations. So all in all, yes, there are extreme differences interregionally throughout the brain in this CS-GAG sulfation patterning. The second question I wanted to answer was, are there intra-regional differences? And this would be between regions within the same general region. And I chose the hippocampus because morphology-wise, it looks as though the PNNs take a different shape based on whether you're in the CA1, 2, 3, or DG. For example, the CA2 in pink forms like a mesh-like formation. The CA3 has a punctated-like formation, and the CA1 and DG show more of extension coverage of the processes. So to perform this analysis, I streamlined a laser capture microdissection coupled liquid chromatography tandem mass spec protocol, which was basically we were able to take each of these very minute and small regions through laser capture microdissection and put them directly into the mass spec pipeline for analysis. And this was done in collaboration with the University of Washington Dirk Molecular Imaging Corps. And also done in collaboration, this initial studies with Dr. Jared Scarlett at the Seattle Children's Hospital and UWMDI. So unfortunately, dermatin was too low of abundance to accurately quantify, but I did get quantification for the 5 main chondroitin sulfate isomers. What we found was that there are vast differences interregionally and intra-regionally now, intra-regionally between the hippocampal subregions, where surprisingly, it's the CA1 that shows the greatest plasticity in the hippocampus, and the dentate gyrus shows the least amount of plasticity. And that is observed by the increase in the developmental C isomer here in the CA1 in gold, and the decrease into the dentate gyrus. And when we took a look at the disulfide isomers, they corroborated this initial finding, where the most plastic neurite extension and circuit formation isomer, the D unit, was highest in the CA1, while the PNN stabilizer isomer, the E unit in red, was highest in the dentate gyrus. I also want to bring attention to species differences, and I'm really glad this was brought up in a few additional talks because there are differences as you move across species. We did the same analysis in rat and found that although the CA2, CA3, and DG showed a very similar pattern as in mouse, there are differences in the CA1 plasticity between these species. However, when we stain for the PNNs and the CS gags with the lectin, we visually see that there's differences in their ability to form stable structures between mouse and rat. And so this finding is not surprising, but more of confirmatory that there are differences between species. So what does this mean in terms of plasticity in the hippocampus? Well, in terms of a direct interpretation of the data, and we apply it to what is known in a simplified model here of the circuitry formation and signaling in the hippocampus, incoming signaling input from the entorhinal cortex enters the DG and follows a very, this is simplistic modeling, but it follows almost a linear synapse formation signaling going from the DG to the CA3, CA2, and CA1, where it then exits the hippocampus back to the entorhinal cortex. So when we overlay our plasticity analysis, we find that the incoming input from the entorhinal cortex is entering a region of the hippocampus that is going to have very stable synapses, and it's not going to be plastic to the outside environment. However, the way this incoming information is processed throughout the hippocampus is it's able to be deciphered differently based on the plasticity of the latter regions. So I'm doing good on time, so I did want to answer one third question. Since this is a Geroscience symposium, as a recap, we were able to show inter and interregional differences. So what happens in advanced aging? How are these changes influenced? And so this is in collaboration with Dr. Arg Logson at the VA, where we compared 12-month control mice, so wild-type mice, with the SAMP8 mouse brain. And the SAMP8 mouse is a mouse model of accelerated aging. And so in this model, we first noted that there are extreme differences in the way the hippocampal CS gags were formed in the SAMP8 model. We knew from a bulk analysis that they were going to have an increase in success. An increase in 4S. But what we did not anticipate was that they were going to lose their plasticity pattern throughout the hippocampus. So compared to the control mouse at the matched age, where the CA1 was most plastic, and then we lose plasticity to the dentate gyrus, the SAMP8 has no plasticity throughout their hippocampus, at least in terms of what the isomers are suggesting. However, we do see that the disulfated units keep the general pattern of plasticity. So similar to the wild type mouse, we found that the highest amount of PNN stabilizing E unit is indeed in the dentate gyrus, similar to wild type, and that there is a higher amount of the neuroid extension and regeneration isomer D unit in the CA1 as in the wild type. So putting together, it really is the 4S and 6S ratio, gold and blue bars, that are changing in the SIMP8 model and not the disulfated units. And then when we look in terms of how this affects plasticity in the brain of the SAMP8 mouse, it suggests that their brain is similar in terms of the decreased plasticity of incoming signal input into dentate gyrus, but that it's unable to the rest of the brain is limited in its plasticity in the imaging, in the information processing, and that this could very well influence the way memories and cognition are developed in this model. So conclusions of this talk and future directions is that I really hope I was able to show you that CS-GAG sulfation patterning is not universal throughout the brain. There is changes, spatial temporal changes throughout neurodevelopment and advanced aging. And these changes are both inter and intra-regionally defined. Some ongoing studies is that our lab is very interested in translating these effects across species as well, like Christine mentioned the importance of this. And so we are well underway in these translational studies in non-human primates and in humans from development into the adulthood in a multi-institutional and departmental collaboration between Seattle Children's Hospital and the University of Washington ADRC. And so from here, I'd like to acknowledge many, many people who are involved in this project, and even more who are not listed on this slide. And I'd also like to thank all my funding sources for their support as we work through these questions. And with that, I'll take your questions. So thank you.
I'm going to start with an online question. It says, I might have missed this, but is there any evidence linking these changes to cognitive function?
Thank you. You can hear me with the mask, right? Okay, cool. So many of the functions in terms of the isomer changes in cognition have to do with if you remove the isomers or you degrade the isomers, then what happens to the cognition? So it's sort of a crude way of asking how important are these particular isomers. If you remove the isomers in vivo, yes, the animals will lose a lot of their memories, particularly that of fear conditioning. So these isomers are absolutely critical in stabilizing the synapses and neurocircuitry involved in memory formation.
Another online question. It emerged about 10 years ago that the SAMP mouse strains arose from introgression of AJ mice kept in the same lab into the alleged strain. So you're looking at genetic polymorphism, including hybrid dysgenesis, rather than rate of aging.
Yes, I think that's a statement. Thank you. Okay.
How do you reconcile these findings with the higher rate of neurogenesis in the dentate? Do newborn neurons lack nets entirely, or do they have more immature isoforms?
I love this question because that's why the results were so surprising to me. I too really thought we were going to get a large plastic environment in the dentate gyrus for that exact reason, the neurogenesis. And we don't. And so that's when we start to think about those exact points, which is it the composition different around specific cellular components than others? And I don't have that data today, but we have been doing single cell mass spec imaging of the different isomer compositions. And I can say it is possible for us to answer that question. I don't have that data for you today, but we can do single cell CS-GAG sulfation patterning at this time.
One more question. Is there a similar pattern of change in the SAP mice with age, maybe accelerated compared to wild type?
So what we looked at was the SAMP8 mouse compared to the wild type at 3, 6, and 12 months of age. We started with the bulk analysis, and at every age that we compared them to, the SAMP8 had a more developmentally mature phenotype in their C-ascatic sulfation patterns compared to the C57. It is then that we decided to look at the intra-regional differences, because the bulk pattern suggested to us that they already had advanced maturity at every developmental age we looked at. I will point out that, again, I think this is important to corroborate against species. And also an earlier talk was to confirm background differences as well. And I think that's very important. And I bring that to your attention because that is something we're also following up, is whether one particular mouse background is more predisposed to have an aging phenotype because of changes in their baseline sulfation patterns. Thank you.
We are going to take a break until 1 for lunch. Oh, yeah.
So the lunches are, can everyone hear me? No. Can everybody hear me? Okay. So the lunches are in the lounge to the right when you come out. They will have your name on them. And then from there, if you didn't order one or forgot to, those are the extra labeled ones. Yeah. Ray or HEA. If you want, we can download it as well, and you can do it from like an actual PowerPoint.